Hello and good morning. My name is Bridgette Gifford, and today I will be presenting predicting SARS-CoV-2 viral load from gene expression data using machine learning. Next slide, please. All right, let's begin with some background. The initial study consisted of over 3,000 Marine recruits, 1,000 of whom tested positive for COVID-19 at some point during the study. The goal was to learn more about the transmission of the coronavirus among young adults. In fact, by studying Marine recruits in their close living environments, we were able to further investigate how COVID transmission might occur on college campuses as well. So, how do we do this? Molecular data and viral load, or how much virus is in the individual system, was collected for everyone involved. Additionally, gene expression was measured in intervals throughout the study, whether it be immediately before or after testing positive. Next slide. With that background in mind, what are the key questions we are trying to answer? First, can we predict, based on gene expression data collected, whether someone has COVID or not? Second, can we predict viral load from gene expression data, in this case, RNA-seq data? Finally, based on this, can we determine which genes are most predictive? Next slide. As I mentioned in the previous slide, we measure gene expression by looking at RNA-seq data. Well, that begs the question, why do we use RNA-seq data? Some of you may be familiar with the central dogma of genetics. In simple terms, it states that DNA is transcribed into RNA, which is then translated into proteins. These proteins, of course, are responsible for keeping us alive. Because DNA codes for our genes in the same proportion, it makes it hard to look for COVID-specific genes. Think of it as looking for a needle in a very big haystack. Instead, we look at RNA. Unlike DNA, RNA translates more of a certain gene based on immediate need. In other words, more of certain genes are expressed at a certain time. This makes it easier to find COVID-specific genes. Finally, if you look to the bottom right, we have a chart that shows a mock-up of how we collected the data. On the far left column, we have a list of genes found in humans. On the top row, we have a list of participants. What you see below is a mock-up of the gene expression data. It is how much, say, participant 1 expresses gene A. Next slide. So now that we understand what it is we're looking for, how do we go about actually making a prediction model? The answer is machine learning. Machine learning algorithms build a model based on sample data called a training set. This is so it can improve and make predictions without being given the real answers. Then after the algorithm is done training, the test set is used as a test to see how well our model performs. The only problem is with large multivariate data sets, it's often hard to summarize or visualize the data in a chart or plot. Therefore, PCA is used to reduce the dimensions of data by finding a new, smaller set of variables that retain the most interesting of the training set's information. Within machine learning, there are two main subsets, supervised and unsupervised learning. Two of my models use supervised learning. Supervised just means that we have some prior knowledge of what the output values of our samples should be. My first model is a classification example of supervised learning using binary data. It predicts either COVID detected or not detected. My second model is a regression example of supervised learning using continuous data. It predicts the amount of someone's viral load based on gene expression. To begin my training, however, I made a model using unsupervised learning to study about the batch effect. I will talk about this on the next slide. Unsupervised learning is just used to learn about the inherent structure of the data without having specific labels. Next slide. As I said before, my first model was created to study the impact of the batch effect on gene expression. To collect the samples, RNA had to be cultivated on special RNA sequence plates. That's what you see in those three columns. In order to check for technical or non-biological variation within the samples, we included data from every individual and labeled it by the sequence plate number it was associated with. Then I used PCA to reduce the number of dimensions of the samples. Finally, I plotted out the results. As you can see, there are no obvious clusters in the data. This means that all the plates were prepared in the same way, and any variation we see will be because of biological variation in the samples, not by variation in sample collection methods. Next slide. After checking for the batch effect, I created my second model, a supervised classification model that predicts whether COVID-19 is detected or not. What you're looking at is called an AUC-ROC curve. AUC is the capability of a classifier to distinguish between classes. The higher the number is to 1, or the higher the curve is to the top left, the more capable the model is. In this case, I plotted both the AUC-ROC from the PCA dataset and from the non-PCA dataset. The two plots showed very little difference, with the non-PCA curve having an AUC of 0.935 and the PCA curve having an AUC of 0.945. This is very good. This means that reducing the dimensions of the data had very little effect on the model's efficacy. It also means that the model can effectively distinguish between the positive class where the virus is detected and the negative class where the virus is not detected. Next slide, please. My next model in my research aimed to predict viral load based on gene expression. In order to study viral load, we look at the CT values, or the cycle threshold. This indicates how much virus an infected person harbors. To compute the CT values, we look at the number of PCR cycles that are necessary to spot the virus. You may be familiar with PCR tests, the method often used to test to see if you're COVID positive or not. One of the most important things to note is that CT values are inversely proportional to viral load. The higher the CT values, the more PCR cycles it takes to find the virus, the lower the actual viral load. Finally, the reason why studying viral load matters is because a higher viral load in an individual means that they are more infectious. Studying CT values can help epidemiologists track outbreaks or identify people of high risk for severe disease. Next slide. So now that we have discussed what CT values are and their importance in viral load, we now look to the results of the viral load prediction model. LASSO is a type of linear regression that utilizes regularization methods. All this means is that the model automates feature selection and elimination, leaving only the most variable and meaningful data. Now, let's look at the plot results. Here, I have made a plot to illustrate the prediction error of the LASSO model. The identity line is what the plot would look like if the model's prediction correctly aligned with the actual CT values determined. The best fit line is how well our model fits to the identity line. As you can see, there's a positive correlation. The predicted viral load follows the general trend of the actual viral load. It predicts relatively low load when it actually is relatively low, and vice versa. However, it is not a perfect model, which is to be expected. Predicting viral load from gene expression alone is a difficult problem with many factors. Still, the model shows that there is significant relevance to studying how involved gene expression is with viral load. Next slide. In addition to the plot of the CT value prediction error, I also made a plot for CT values residuals. This is essentially showing the identity line from the last plot, except this time it is on the x equals 0 line. Above the line, we see how much the training set and testing set overshoots the actual CT values. Below the line, we see how much the training and testing set undershoots the actual CT values. As you can see, the points are evenly distributed with no specific pattern. We can therefore conclude that there is no bias in either direction. After all, we wouldn't want a model that consistently over or undershoots. Finally, we should note that our model still has some overfitting. While the R squared value of the test set is 0.302, the training set's R squared value is 0.57. While this is not overly significant, it should be taken into consideration when moving forward. Next slide, please. Finally, the last question we aimed to answer was, which genes are most predictive of viral load? From the LASSO regressor, we found that 48 genes had non-zero coefficients. Among them, 2 genes stood out in particular, LY6E and IFI44L. Both of these genes were linked to COVID-19 disease processes in previous studies. IFI44L codes for an interferon protein that acts to inhibit viral replication, while LY6E codes for lymphocyte antigens. Lymphocytes are just immune cells made in bone marrow, and LY6E acts to restrict the entry of the SARS-CoV-2 into the cell. Looking at the plots, we see that when CT values are high, gene expression is low. This makes sense. A high CT value means a low viral load. It would make sense that the less virus one person has in their system, the lower their gene expression is to fight the infection. Next slide. Now for the conclusions. We were able to successfully create 2 models based on gene expression alone. The first was a classification model that predicts COVID-19 positivity or negativity. The second was a regression model that predicts the amount of viral load. From the lasso regression model, we were able to isolate 48 genes that showed the highest feature significance. 2 of them were LY6E and IFI44L that were previously associated with COVID-19 studies. Finally, we can conclude that the studies in viral load in COVID-19 patients should be continued, as we see a lot of significance in the data. Next slide, please. Before I go, I want to give a huge thank you to my wonderful mentors, Dr. Rachel Silfan and Dr. Natalie Sauerwald. Their constant support and openness to me really made my research experience an enjoyable one. I also want to thank Dr. Olga Troinskaya and her lab team for giving me access to gene expression data, as well as allowing me to sit in on some of their weekly meetings. And of course, I want to thank Dr. Casey Wagner for being an advisor and mentor to me during this whole 8-week adventure. Finally, I want to thank the Simons-NSBP Scholars Program for giving me this wonderful opportunity. Next slide. Thank you for your time.
So there's time for a question or two if anybody has them. All right, yeah.
Just, do you have a sense if you were to take these techniques and apply them to a different virus, say the one that causes flu, would it be helpful?
Could you hear that, Bridget?
Yes, I can hear that.
Okay, go ahead.
I think it's definitely possible. The techniques are the same. All you need are the gene expression data to input into the model, and it should run the same way. So I definitely think it could be used in a variety of different viruses.